Okay, so today we are going to be digging more in detail into RNAi mechanisms, looking at a paper here that looks at RNAi mechanisms as mechanisms for antifungal resistance. Is this working? Let me just check. Yes, the microphone is on. Okay. The paper is broad antifungal resistance mediated by RNAi-dependent epimutation in the basal human fungal pathogen Mucor circinelloides. Again, like I'm not a fungal guy, but I've worked a lot on yeast, so much of this paper I could reasonably understand, but I did have to do a little bit of research to understand some of the background of this. Okay, so there is a disease, Mucormycosis. So it's an infection with a fungus. So I mean, that's probably worth just talking a little bit about infections with fungus. Does anybody like what makes infections with fungi different from other infections in the sense of how you treat them? Yes. Yes. Okay. So what was said is that fungi are eukaryotes and humans are essentially like derivative of ancestrally some fungus at some point. So we're very closely related. And so medicines that affect fungi often have high toxicity in humans. So it's difficult to get rid of fungal infections as opposed to maybe like a bacterial infection where you can give like an antibiotic. So for that context, there's that. In general, we have an immune system that fights off fungal infections pretty well. But you do see fungal infections in people who have suppressed immune systems. So if you just get like a transplant of a new organ, or the doctor might put you on immunosuppressants, and then those people can often get infected with fungi. Also, people who are older or younger, very, very young or very, very old, have suppressed immune systems. And so those people are sometimes at risk for fungal infections. So this is one of those things that apparently infects and destroys your face. So it gets into your sinuses, your eyes, your brain, and creates tissue damage. And it's can be difficult to get rid of. Okay. So that's this particular disease. Let me see as I go through this, anything else worth mentioning. The infectious genera are Rhizopus, Mucor, and Lichthemia. Let's see. Okay, so let's talk about this concept, which is the premise of the paper, which is epimutations. This is a new word for me. I had never seen this word in the genetic literature, perhaps because I'm not in this field or had not researched this topic. But who read the paper a little bit and can describe for me what epimutation means? What do you mean by structure? That's not how I interpreted it. Any other? That's reasonable, but I feel like you guys are not hitting the main point. What? That's probably closer. Like that could probably be a mechanism of methylation could be a mechanism perhaps of epimutations. What I gleaned from it, you guys tell me if you agree or disagree. So a classical mutation, right, would be like gene X, and the actual gene is in the chromosome and acquires some mutation. Maybe it's here, and then maybe you get zero transcription, and that would be like a knockout. That would be a classical mutation. What I thought epimutation is, what I think it is it's not a classical destruction or change on the actual chromosome. It's just something that leads to changes in transcription. So you could have a totally functional gene X, and something might lead to changes in the transcript, which would then be an epimutation, which then shuts off the gene. So that obviously makes sense in the context of RNAi, where RNA interference, if there was an RNA interference pathway, will somebody open the door? If there was an RNA interference pathway targeting one of these gene X's, that could lead to suppression then of the transcript, and that might lead to some downstream phenotype. In this case, the phenotype being fungal resistance against these drugs. Do you agree or disagree? Okay. Okay. So hopefully I got that somewhat right. Okay. Okay. So that's epi mutations. Um, let's see. So in this case, the premise, again, the premise is imagine if this is some gene X, right, that is making transcript in this organism, in this fungal organism, there's going to be a gene for dicer, and there's going to be a gene for the risk complex. And there's going to be, here's the interesting thing is how the small RNAs get expressed. That's not defined in this paper, but at some point, some double-stranded RNA product gets expressed that matches the gene X, and then dicer will chop it up, the risk complex will grab onto it, and then make it single-stranded so that it can then target the transcript for degradation through Argonaut. So we've already discussed dicer, risk complex, Argonaut, the endonuclease targeting the messenger RNA and that mechanism. So none of that is new. The only thing that's new is that this is now in a fungus, and the outcome phenotype is not insecticide, but antifungal resistance. Okay, other stuff that's worth talking about. The paper focuses a lot on this drug, which is called 5-FOA. This is worth knowing what it is because it's a very common reagent in yeast and in this case, fungal genetics. So 5-FOA, that's used as a counter-selective agent against URA3. So let's also quick define selection versus counter-selection. This is the first time we've introduced counter-selection. And it's quite actually a very important topic for synthetic biology, but because it's a little bit more advanced, I don't often introduce it. So selection, give me some examples of selection. Perhaps you have an ampicillin resistance marker gene that provides resistance against ampicillin. That's classical selection, where you're selecting for the presence of this cassette in a plasmid. Okay, so classical selection is you're selecting, it's like, this is called positive selection, selecting for something. So then counter selection is what? Take a guess. Yes, selecting against, you could phrase it as selecting for the absence, yes. Selecting for absence of something. And so one of the most common reagents here that they talk about is this 5-FOA. If you put 5-FOA in media that yeast are in, if they have a plasmid with a URA3 oxytrophic marker, so that means they can produce uracil. If they have that, the product, the uracil synthesis pathway, can take this chemical and turn it into a toxic byproduct. And so essentially, if you have a functional uracil synthesis pathway, you die in the presence of this drug because it takes this drug and it converts it into a toxic substance. So if you had a plasmid in a yeast that had a URA3 cassette and you put 5-FOA in the media, what will happen to the plasmid? It's already in the cell. So rephrase it. Your thinking is correct, but rephrase the wording a little bit different. Not that it won't be accepted, but rather it will. So the plasmid is already in the yeast budding cell, and there's this drug in the media. What does the yeast cell have to do in order to survive? Yeah, it's got to get rid of the plasmid. That's a better way to say it. It'll, or you might just say like it'll spit out the plasmid. So it either the cell will divide and then it will select for the daughter cell that does not inherit the plasmid. That would be like one way of spitting out the plasmid, or perhaps the plasmid itself might acquire some mutations that shut off the gene. But it's essentially selecting for the absence of a functional uracil synthesis pathway. So this is commonly used in yeast genetics, where people will do insertions and then they'll swap in and out things. So for example, let's see if I could draw this out. If you might create some kind of an insert. So imagine you have like a homology. Remember, homologous recombination works really well in yeast. Let's say you have a homology region 1, homology region 2. Let's say you have an insert with gene X, and let's say the selectable marker that you're selecting for is UR3. And let's say you could recombine this onto the chromosome and you create some strain. That might be step one, to insert this thing. And then oftentimes in science, you want to test what happens without it. So to get rid of this insert, you can add FOA, and oftentimes that will select for this insert to pop back out. So it's often used in biotechnology and synthetic biology as a way to pop things out that you just put in as to create kind of like negative controls, or to create weird selection regimes where you want to select for specific things to get rearranged on the chromosome. So we all kind of should understand that this chemical is an important biochemical reagent and it's used in counter selection. You should kind of understand what the idea of counter selection is. Okay, so now let's go a little bit more into this. Let me grab this, move this over here. So the paper is not actually dealing with counter selection against UR3. It's looking at these other genes in the pathway. So one of these genes is called PYRF, and another one is called PYRG, okay? And like, I'm not an expert on these genes. You don't need to be an expert on these genes. Just understand that these genes are in what biosynthetic pathway? Well, this is not an inherent pathway. This is kind of like a weird drug that is a mimic of a key biochemical, not metabolite, but chemical, which is called uracil, right? So understand that these genes, essentially there's some synthesis pathway that includes a few genes. These genes are in the synthesis pathway that leads to the production of uracil, okay? So just like UR3, these genes are in that pathway, okay? So if they're in that pathway and you add FOA, the 5-FOA, the selection is going to select against these genes, right? You're going to select for either the destruction of these genes or somehow the loss of these genes, or in the weird mechanism that they observed here, suppression of the transcription of those genes. So what's actually happening in the mechanistic details of this paper is that what's happening is when they add this chemical, it's selecting for this RNAi pathway to turn on and target these genes to suppress these genes. So the interesting thing about this, this is like a reason. So then what this is, what they're billing this as is they're billing this as resistance against this drug 5-FOA. So they're evolving resistance against 5-FOA, but not through chromosomal mutations and not through deletions, but through RNAi silencing of these genes, which then knock down the transcripts temporarily, which allows them to survive in the presence of FOA. Okay, so the interesting thing about this resistance is it's temporary, and it's I don't know if you'd say it's non-genetic. I would say it's non-structurally genetic. Structurally genetic. So in that sense, I did think this was an interesting paper. I had never seen this type of mechanism before. So it's interesting to see a mechanism that it can create resistance through a unique pathway. Are there any questions about sort of like this premise or this idea? Oh, that's an interesting question. You'd have to look into the specifics of what these genes, yeah, like maybe, but it's definitely not lethal because they're able to survive with them temporarily suppressed. So that's my 2 cents on that. But you might have to fact check on that, look a little bit more detailed. Yeah, that's a good question. So yeah, in this paper, they do kind of like these series of experiments where they provide like the bolus of FOA, and then they do more experiments where they continually passage in the absence of FOA, and then the resistance goes away, disappears. So it's kind of like a temporary thing. Okay, history of RNAi in fungi. Let's see if there's anything here that's worth talking about. The first description of RNAi in fungi was quelling, a mechanism for silencing repetitive sequences and transposons in the model fungus Neurospora crassa. So what does this remind us of? It's just an interesting historical note. So they're saying that in fungi, the first identification of this RNAi mechanism was through silencing of transposons. What is that historically similar to? Yeah, it's worth bringing up the analogy here to the P elements because that's the exact same way that RNAi was essentially found in, or at least a P-way pathway was discovered in Drosophila. So transposons are obviously kind of like another separate genetic like enemy of organisms. They're parasitic DNA. And obviously the RNAi pathway has strong selection to inhibit the jumping of transposons in all different organisms, not just fruit flies. And it says, in fact, exactly that. It says, since then, this pathway has been shown to be conserved throughout many eukaryotic lineages, including fungi, etc. So essentially, the thing you want to take home here is that the RNAi, we talked about it in the context of bugs, but RNAi pathways are essentially in like all eukaryotes. They're in fungi, they're in animals, they're in plants, and they suppress various things, commonly transposons, sometimes viruses, and in this case, sometimes they can help evolve resistance. Okay, we talked about the epimutation. Let's see, is anything worth talking about here? It just says that this some of these genes have, or some of these, this mechanism has been studied and found to be a mechanism that can evolve resistance against some other chemicals, not just the 5-FOA. That's the context of the field. Okay, anything else? Okay. Shut that phone off, or I'll kick you out. Okay, this is an important thing that's very important for this study. So it says here, this is in the introduction, or I can't remember if it's in the introduction or the methods, but it says, I pulled this out, it says, to increase the possibility of isolating epimutants, we performed initial screens in 2 RNAi mutant backgrounds. So they give these 2 genotypes. One is deletion of RDRP1, and the second one is deletion of RDRP3. Okay, so they're working, what they're telling you is that in this paper, they're specifically working with 2 specific genotypes, this genotype and this genotype. What are these genes and why is that important in this paper? Did anybody catch that? Yeah, RDRP. Very good. Okay, so this is these are common RNA polymerases. So RNA polymerase 1, RNA polymerase 3. Okay. So, and they're saying they're working in these backgrounds, and they're saying it's more likely, let me just make sure I get the language right, to increase the possibility. So it's more likely to isolate these epimutants that are shutting down those 2 genes in the context of RNA polymerase knockouts. Why would that be true? Well, so yes, so you're right, but let me just try to clarify a little bit more if I can do like a visual diagram. So imagine you have this PYR, what was it, PYRF, PYRG? Somebody help me out here. It was F. Okay, so one of these genes that was being silenced was PYRF, and the silencing leads to the phenotype that they become FOA resistant. Okay, so if you silence the transcript for PYRF, you get FOA resistance. But if you were in a wild type background, that had RDRP1 or a functional copy of RDRP3, they're saying you do not get that resistance. What would be the mechanism for that? Why would you not get that resistance? And they actually didn't know this to begin with. They found this out through their study. So if they're saying, if you have functional RNA polymerases, we don't see this effect. How would a functional RNA polymerase combat silencing? You're getting close, but I would not phrase it like that. When you say get to it, get to what? Well, the polymerase is making the mRNA. So you're like really close, but you're not quite precisely saying exactly the real mechanism for why. So remember that the resistance, two processes that are happening here. Imagine this gene gets made into messenger RNA. Okay. If you have the RNA, you die. If you silence the RNA, you evolve resistance. So the silencing is effectively trying to cut down to destroy the transcript. If you have a functional RNA polymerase, that's like a counteractive process that's going to make more transcript, right? So if you have highly active RNA polymerases, you're going to make more transcripts in general, right? And so it's going to be harder to silence those genes if the context of them having more transcript. Does that make sense? This is actually, it's a difficult paper to understand, but okay, what they're saying is that you can't select for this resistance unless you have knockouts of the RNA polymerases, because if you try to select for the resistance in wild type strains, the RNA polymerases just keep making transcript, and so you can't suppress it enough to get resistance against the lethality of FOA. It's a very convoluted thing to try to explain. Hopefully you guys kind of got that. Yes? No? Okay. But just understand, here's the caveat of this is, and I'm not trying to be super critical of this paper. Like, it's a good paper, but does anybody think that, like, can anybody extrapolate how this might be a criticism of the paper? Yes. So this might not actually be, like, I don't know. Again, I'm not an expert in this field. This might not actually be a real thing that happens in the wild, because they're finding these weird phenotypes in these weird knockout mutants. Does that make sense? So this is not something that you observe in, like, wild type yeast. This is something that they're detecting in these weird knockouts, and they have to produce these knockouts to produce this weird effect. So well, I don't know if the goal here, I don't think the goal here is to develop fungal, like, antifungal mechanisms. Like, their goal is not to develop new medicines, I don't think. I think their goal is just, like, to figure out, to understand mechanisms of resistance in fungi in general. And they're saying they're kind of like saying, like, we uncovered this weird mechanism of antifungal resistance. But the catch is, you never actually see it in wild fungi. You only see it in these weird knockouts. So that's what I'm saying. That's a criticism of this study, is they're producing, like, this weird phenotype that doesn't probably occur in wild fungi. Well, okay, so now, like, since we're bringing that up, and I'm not here to be super critical of people's papers. They actually bring this up in the discussion, and I pulled out so they're very honest about it. What they say, let's see, here's the paragraph right here. Let me just read it. It is possible that these mutant backgrounds are required. So they literally acknowledge in the discussion, it's possible that these mutant backgrounds are literally required to get this resistance. So they're essentially saying, like, this probably would never happen in the wild. I mean, I'm saying a little bit more harshly. They're trying to say it in a way where they can get the paper accepted. But that's, I think, is actually the biggest criticism of this paper, is this might be like a weird phenotype that only occurs in the lab that they've developed. That's an interesting question. Does it make the paper kind of pointless? I think no, because I think it's reasonable to assume that RNA silencing could cause some kind of phenotypic effect. That's essentially what they're saying at the broader level, is that they're essentially saying at the broader level, not everything can be sourced to genetic DNA mutations. There are things like epigenetics that might be just as important for evolving resistance. And I think they're kind of doing a proof of principle here, showing you, like, that can happen. Okay, so that's some, like, intense nuance about this paper that made this paper a little bit difficult. Okay, let's look at figure 1. I actually, again, like, I thought this was pretty hard to understand, but I did my best here. Okay, so in this first figure, again, let's see if I can understand this. First, let's think through A and B. What are these, and what are they looking at? These types of data. Did anybody see what they are? It is some kind of a gel. Okay, so yes, I agree. It's some kind of a gel. But it's not just like classical ethidium bromide. Did anybody go a little bit deeper? What do they actually say? So did anybody see this? It's a blot. It's a blot. So it's not just a gel, but rather it's a gel that utilizes blotting. Okay. So in the case of the only blotting, I guess we've defined is Western blot. Western blot uses an antibody to detect a protein. Okay. This is not a Western blot. Okay. This is a nucleic acid blot. So you might have heard of Northern blots or Southern blots. I can't remember which is which. One of those is DNA, one of those is RNA. They're doing the one that blots against RNA. So essentially the point is that they have probes. They run their gel, they add their probes, and then they're using fluorescence of those probes to detect presence or absence of a very specific nucleic acid. So just understand when you look at this data, what they're showing you is presence or absence of a very specific nucleic acid. In this case, a little chunk of this gene PYRF that's about 23 base pairs long. And in this case, PYRG, a little chunk of PYRG. Okay. So actually what they're detecting here is, quote unquote, if you read it carefully, antisense transcript. So remember, to silence those genes, you need to produce the antisense little chunk, okay, that initiates the RNAi. So when you see this here, and that yellow is not coming through very well. When you see that here and here, this is the R. Again, you have to decode all this stuff, which makes it real difficult to read. The R stands for resistant. So in the FOA resistant clones, what do you see the presence of an antisense transcript that's targeting the gene. So that's them proving, like, mechanistically they can see these little chunks of these transcripts in the resistant. P5, that's 5 passages in the absence of FOA. And what happens to the transcript in the absence of FOA? It goes away. So it's gone. So that suggests, again, this is proving that the effect is transient. It kind of like shuts off. And all that information is sort of encoded in this little blot here. Okay. So E1, again, looking at this, it's a little bit difficult to understand. E1, E2, E3, E4, E5, these are the names of their differently isolated resistant clones. So they're testing the presence or absence of these transcripts in these various resistant clones, okay? And one of these clones is resistant because it's suppressing the PYRG gene. Which one is that? Yes, E4. So E4 is the only clone that is resistant because it's suppressing the PYRG gene as opposed to the other ones. All the other ones are suppressing the PYRF. So essentially what the conclusion from that is that there are 2 separate mechanisms to evolve resistance against this FOA because it's targeting either one or the other genes in the particular pathway to suppress it. Okay, that makes sense. Let's see, what else is worth pointing out? What is this, and what's the purpose of this bottom gel, 5S-RNA? Yes, it's a loading control to make sure that if you have nothing here, or here's a example of a gel where there's literally nothing there in the data. In order for us to conclude that, yes, these really don't have the antisense transcripts, they have to show the loading control to make sure that there is actually like RNA in the sample. Okay, so that's what the loading control does. So, okay, we agree with their premise that it's possible that the resistance conferred from this mechanism is due to the production of these antisense transcripts. Okay. Are there any questions about A and B, or should we move on? Okay. So now this is the phenotypic results of those same, or a subset of those clones. So they analyze E2 and E4, probably because those are the 2 separate mechanisms. Remember, E4 is the one in the PYRG silencing, and E2 is silencing the PYRF gene. Okay, so then you need to look here to start to understand this figure. I'm trying to do this the best I can. You have to look at this little code that tells you what these are, okay? And there's 3 different types of media, okay? So let's first just look at the first media. Okay, MMC, did anybody look up or know what that is? if you see MM in microbiological papers, it typically stands for minimal media. It's typically like a normal concoction that kind of has the minimal amounts of things to be able to grow. So this is minimal media, okay? And so what do you think they're demonstrating by testing on minimal media? These various strains. Yeah, this is just a control for like how well do these strains grow in a normal like media. That's essentially what's happening here. Okay. And then so if you look at the growth on these, okay, you see these ones grow pretty well. And these ones up here, I don't want to make this so colorful that it makes it hard to read, but these ones you can easily see they don't grow as well. Is that correct? Okay, and those ones, what are the genotypes of those? One of those is a natural deletion of PYRG, so they're using that as a control for FOA resistance. So it's got a natural deletion, and because it has that deletion, it doesn't grow very well on normal media. So that actually says what you said, Thomas. That like again, what do I want to say? There's a fitness cost to acquiring these mutations. That's validated here. Okay. And then let's see here. I can't remember what P stood for. I'm writing here that that was a negative control. I can't remember what that strain is. Somebody try to look at that. But you see the same pattern here. Okay. So let's move to the media on the far right. So we'll look at here on the far right. This is the MMC, so it's the minimal media plus the uracil plus the drug that'll kill you if you're making uracil. Okay. So in this case, you would only be able, on the far right, you can only grow in these plates if you are resistant against the FOA drug. Okay. And so you can see here that the E2, which is the top right, this one, that one grows as a resistant clone in this media, and the E4 grows as a resistant clone in this media. So these two dots, the conclusion that the fact that these two dots are growing means that the E2 and the E4 are resistant against, I'm writing here bad, but are resistant against 5-FOA. This is actually the data that proves that those things are resistant. And remember the deletion of PYRG and the deletion of PYRG here. These are the controls, the positive controls for resistance. So these is also the same conclusion. These are the positive controls that are resistant. And so because this mutant looks like the positive control, it's resistant. Okay. Let's look at this. I'm trying to figure out what that P is. Parental. Okay, so that's the wild type. Then I would imagine that's the type that these were derived from. So this just shows you that the parental is not by itself resistant. So that mutation had to be acquired. Ditto here, the parental strain is not by itself resistant, so that mutation had to be acquired. Okay, so it's kind of like essentially the gist. Complicated figure, difficult to understand, difficult to decode, but the essential gist of this paper or this figure here is that on the bottom, they're proving that the mutants that they acquired are actually resistant against the FOA drug. And then up top in A and B, they're hypothesizing that the reason they're resistant is because of the production of these unique antisense transcripts. Okay, so that's that figure. We kind of did the conclusions. We'll go into this question in a second. Okay, so then they go into this next. I actually thought this was a really interesting figure. I always like seeing data displayed in a way I have not seen it quite before. So here's the figure 2, I think. So in figure 2, what are they measuring? So if you're ever presented with a graph, the immediate first step, even if it's confusing, is to look at the axes. What do you see on the axes? Okay, so on the y-axis is number of reads. So this is obviously a sequencing reaction. So it's going to be a high throughput sequencing reaction. What are they actually sequencing? Yeah, they're sequencing the small RNAs. So this is actually a really interesting experiment because they did a high throughput Illumina sequencing on a subset of RNAs called small RNAs. So that means they had a purification process to purify specifically small RNAs, and then they sequence all small RNAs from the entire that are being expressed from the entire genome of all those cells. So they actually had to prepare. I looked at the methods how they do this, just to, I guess, see the details. So they prepared they extracted small RNAs. They went through a purification process, and then they prepared libraries that were sequenced. So they cloned these small RNAs into plasmids, and then they sequenced those, okay? And then they can quantify essentially like the abundance of those via the reads, okay? So that's what's on the y-axis, is essentially the quantity, the number of these small RNAs. Now, what's on the x-axis? What is the x-axis? Yes. So this is a base pair in the genome. So when I draw my genomes, I draw it like this. Imagine this on this x-axis is actually just a length of the genome. And this sequence right here they're annotating it. This is actually the sequence of PYRF, that gene that's being silenced. So if we were to draw it with my symbolism, which they should use my symbolism, they would the start codon goes here, and the gene looks like this. Okay, so it's going that way. There's the stop codon, there's the start codon. So all the interesting information is going to be between here and here. Okay. What do you observe from the data between here and here of this reads of small RNA that correspond to exact sequence that's antisense, or that's sequence that matches this gene? What do you observe? Yes. So yes, very good. So this is the red is the silenced. That's the resistant. You see a whole bunch of these peaks. So you're seeing read pileup in small RNAs that correspond to this gene. So that's already like something wacky is going on here, where in other parts of the genome, it's not expressing these small RNAs, but right here, it's expressing small RNAs that match these sequences. Okay. The other thing to notice is that some sequences go above 0 and some sequences go below 0. So what does that mean? Well, you can't have, so that's what I originally thought when I looked at this, and then I had to very carefully read the legend to understand what it was. So the reason it's not transcript reduction, you can't have transcript below 0. So this is not actually, so below this 0, it's not actually like reduction of transcript. Did anybody else catch what it actually is? This is what made this figure actually difficult to understand. You really had to kind of like figure this out. It's here, and it's also, yes. So again, this figure is actually kind of hard to decode, but below the 0 is an abundance pileup of antisense. Excuse me, hang on. Is an abundance pileup of antisense transcript right here. So this sequence right here. What it's detecting is it's detecting an antisense sequence pileup for a sequence that matches right there. Okay. And this is pretty much the only antisense transcript that's piling up. So all the silencing is probably from this thing right there. Okay. Now in the other gene, this is from a different clone, silenced. They see a similar pattern in that they're seeing, but much more abundantly, they're seeing much more abundant read pileup of antisense transcripts. So the silencing data for this gene, the PYRG, is actually much stronger in the E4 clone. The data is much stronger in this one. They're getting much more abundance of antisense transcripts. Okay, so kind of a weird figure, but once you kind of understand the decoding, it's not too difficult to understand. Essentially, the main take home from this is that according to the sequencing data, they are detecting small antisense RNA transcripts that match the antisense of these genes, which can correspond to how they're being silenced. Okay, are there any questions or disagreements with that? Okay, that's essentially like the take home from that piece of data. Let's see. Here's what I had a question about. Oh, maybe. here's what I had a question about, which was, how do these products get made? So imagine you have your gene, let's say it's PYR. Let's do G. Let's do G. PYRG already has like some innate promoter. And what I don't understand about this paper is all of a sudden, like you select for this increased production of antisense tiny transcripts right here. So essentially what's happening is some kind of an RNA polymerase is coming on like in the middle of the gene, like at these spots. So maybe there's like one here, and one here. And these RNA polymerases are coming on in theory, and they're transcribing like tiny antisense messages, and then they're just stopping transcription and they keep coming back to that site and they keep making this. So I don't know how, like, mechanistically you select for that. I don't know what that is or how that gets built in. Like maybe you select for a mutation that creates a reverse promoter inside the gene. Maybe that's what's happening. I have no idea. But actually that would be a change in the DNA. So they could not be that. I don't know. That's what I could not understand essentially about this paper. That's what I'm pointing out to. Like the difficulty for me, I cannot envision mechanistically how this happens. How all of a sudden you get like a production of these tiny messenger RNAs that correspond to that spot highly selectively. I have no idea how you genetically produce that, but I'd be very curious to know the mechanism on that. So in the follow-up paper, that's what I'd try to ask them to figure out, is I'd try to ask them to figure out, like, how does this actually even happen? And if you discovered the mechanism of how it happens, you might get some really interesting biology there. Okay. This will probably be the last, second to last figure we talk about. So in this, what they say is that small RNAs have a prevalence of a terminal uracil. So if you're making these tiny antisense messenger RNAs, it just so happens that more commonly the last base is uracil. And so here they're using this as like stronger evidence to build their case that these antisense messages are getting built up. Where if you look at, let's see here, the reads, if you look at those reads that are targeting those genes, the vast majority of them end with a U. And so they argue that that is evidence that supports that these are small RNAs. Again, they're trying to build the case that these small RNAs are the reason that the resistance is occurring. So that's essentially the gist of A and B, except A is the first gene, F, and the bottom one is G, the other gene. Okay, figure 4, then we'll wrap it up. Let's look at this. This is just the final sort of proof that the gene is actually getting silenced, I think. Yeah, so this is PYRF expression and PYRG expression. Here is some kind of a positive control. So in this positive control, they're getting like normal levels of this gene expression. In the mutant, they're getting what would you call this according to the RNAi lecture? So when I gave the RNA lecture, I said, what? Yeah, this is a knockdown. This is actually the knockdown. So actually, when I gave that lecture, I said, you'll see figures that look like this. Here's where you actually see a figure that's showing you the knockdown. So it's showing you that this gene's expression goes down in the mutant. Ditto in this mutant, E2. What are the revertants? Yes, so the way that they do this is they remove FOA from the media, and they continue to pass the microbe on that media without FOA. So now the counter selection has disappeared. And then they revert back to the original phenotype, and you see the expression of these important genes go back up. So they're telling you, again, they're proving here that this is like a transient effect that transiently happens to confer resistance, and then they can revert back to the wild type. And then you kind of just see the rest. The rest is just similar stuff. Okay, so that's the gist of the paper. Let's see, is there anything else worth talking about in the discussion? I mean, probably what's worth discussing is why have a mechanism like this? This is an open question. Like I'm brainstorming myself. Why have a mechanism for resistance like this? Like if you're a biological organism, how could this be useful? As opposed to a different type. So remember, a different way that you could evolve resistance for this is they could just knock out or mutate their own P, Y, R, F, or G genes. So you could evolve resistance through either the silencing or the knockout. And this is a mechanism that's subtly different. So what's the advantage of this? Why have this? Why not just evolve resistance classically the way that everybody understands where you mutate the DNA? Yeah. Yeah, there might be a real strong selection against actually mutating the DNA of those genes. And so this might create a very unique situation where you if you're faced with a choice of knockout an essential gene or die, it might select for an intermediate, which is kind of like this, like where you can not die, but you can also get resistance in a temporary silencing mechanism. So this might confer a temporary ability to evolve resistance in genes that are essential for function. I think that's kind of the best hypothesis, and I think they kind of say that. Where do they say that? Yeah, it says serving as it's a bet hedging strategy. That's what they say in the discussion. They say this mechanism might serve as like a bet hedging strategy where you don't want to put all if you completely knock out the gene, you're kind of putting all your eggs in that basket. But if you're kind of just developing a way to transiently shut it down, it's kind of like an intermediate. And so you kind of are the best of both worlds. So it's a bet hedging strategy that enables rapid and reversible responses to a variety of environmental conditions. Okay. So that's kind of the gist. Any final questions? This paper was okay. I thought it was kind of hard to read, kind of a niche mechanism, but I thought it was pretty good. Okay, we'll end it there. Thank you.